Hello everyone, and welcome to my short poster presentation for Animal Behavior Live 2023. My name is Anna Kawamoto, and I'm very happy to present to you my study on visual acuity in the common wall lizard, aka Podarcis muralis. In the realm of animal communication, visual signals play a pivotal role, and to better understand how animals perceive and respond to these signals, investigating visual acuity is crucial, as you will notice in my study. But before delving into my presentation, let's just begin with some general definitions to give you the context of the study, starting with signal, which is any act or structure that affects the behavior of other organisms, evolved and maintained because of those effects, and is effective because it makes information available to receivers. The species I'm particularly interested in is Podarcis muralis, a small lacertid that uses various visual signals, including dynamic displays, such as foot shakes, and conspicuous color patches, because yes, indeed, it shows on its flanks UV blue patches that are considered chromatic signals. So why the visual acuity of this little lizard is such a compelling subject to explore? Well, there are multiple facets of the biology and visual communication tied to their diurnal activity, preference for open habitats, territoriality of males, male-female interactions, but mostly predation on small and fast-moving prey, as well as the ability to detect predators at considerable distances. And on top of these, as I've mentioned previously, the peculiar chromatic signal this species demonstrates heavily rely on vision and require the ability to distinguish fine details. Since visual acuity is the ability to resolve static spatial details in the surroundings, estimated will provide valuable insight into the species' communication and survival strategies. Expressed in cycles per degree, it's the number of discriminable black and white stripes within a single degree of visual angle, and I will better illustrate this definition through my experiments later on, but briefly, visual acuity is often estimated behaviourally or histologically, and our study aimed to estimate it by adopting this dual approach. So because vision relies on the projection of an object image onto the retina, its effectiveness is constrained by several ocular parameters, such as the density of photoreceptors. So here we sought to obtain a retinal ganglion cell topography to estimate the visual acuity histologically. So we prepared a retina Hall mount to count the number of these cells and made the topographic distribution map. And we searched for any area showing a particularly high amount of these cells, such as a fovea or a visual streak. Concerning our behavioral experiment, we've constructed an optomotor apparatus consisting of a rotating drum with black and white vertical stripes on its inner wall and in which we place the animal in its center. We changed the width of the stripes depending on the CPD we wanted to test, and by rotating, as you can see in the video below, this apparatus induces a reflex-orienting-movement called optomotor response, allowing us to estimate the visual acuity behaviorally. Moving on to the results, our histological analysis revealed a retinal ganglion cell peak density exceeding 7,000 cells per millimeter square in the central temporal area of the retina, corresponding to an acuity of 1.1 CPD. We've obtained similar results from our optomotor assays as a high amount of positive optomotor response for 1 CPD, drastically dropping from 2 CPD, and these behaviors were rapidly elicited for 1 CPD, while we found an important latency for 2 CPD. We've calculated the minimum separable angle as well, which is an equivalent of the acuity, considering only the parameters of the drum and the distance of the animal's eyes to the stimulus, and we've obtained a value of 1.73 CPD. So to conclude, our dual approach estimated an acuity between 1 and 2 CPD for this species, but beyond these acuity values, what does this mean in a functional context? Well, it implies that an individual would be able to detect a conspecific from almost 10 meters away and perceive a 2 millimeter long UV blue patch from 40 centimeters away, as well as any prey of the same size. And beyond mere detectability, we were also interested to get more details about discriminability. So we were generating images illustrating what this species would resolve in its surroundings, such as its UV blue patches, at different distances in the visible and the UV range. And with details on the blurriness, as you can see in the left column, UV blue patches are more or less discriminable for 10 centimeters, but blurry for 1 meter. So it seems that this species can visually detect objects from quite far away, but details are resolved at shorter distances, emphasizing the role of these chromatic signals in close-range communication. And I want to thank my supervisors and collaborators, and please ask any questions.